Thank you very much for the nice introduction. So it is not a secret society. We are still working out on the membership fees. But as soon as this is resolved, you will all know about it. But in a nutshell, the reason why we created the society is because there is a big reproducibility crisis in many subfields of biomedical science, and bioinformatics is actually one of the fields where we can do tremendous good in terms of making all the analysis easier to understand, easier to reproduce, and easier to reuse. So I'll touch upon that in my last slides if I manage to get there. Okay, so I would like to thank the first two speakers. They introduced a lot of concepts that I'm going to use in my presentation. So here, I'm not going to tackle the prognostic biomarkers that much, which is more the predictions of the natural evolution of the disease, either with a treatment or with standard of care. I will focus today on therapeutics, drug response prediction. So we have been extremely quite good in the last decades to actually define what makes a tumor. What are the hallmarks of cancer? What are the pathways that need to be activated for cancer to actually grow? And here you see the hallmarks, the famous hallmarks of cancer from Hanahan and Weinberg. And what I would like to draw your attention is that for all of those hallmarks, we actually have developed so-called targeted therapies. So we try to understand what are the crucial pieces for tumors and try to target them with drug and different chemical compounds. So here is a non-exhaustive list of those inhibitors targeting specific hallmarks. So you would think that the cancer would be cured by now because we identified all the hallmarks and we have all the different chemical compounds to target those hallmarks. But of course, life is not that easy. Even for given tumor type, there is a lot of inter-patient heterogeneity, but also intra-tumor heterogeneity, different clones of cancer cells. They depend on different kind of pathways. So even with targeted therapies that kill the bulk of the tumor, you may still be cured because a small clone of that tumor is resistant to that drug. So the problem here is that you would like to identify those patients that are more likely to respond to any given therapy. And the big issue is that, that's why I was asking about the definition of actionable mutations. Here I'm gonna call actionable something about the treatment. So an aberration that you find in a tumor that could be used to predict which therapy we should give to that patient. And the problem is that if you look at the genotype match clinical trials out there where we profile the tumors, mostly using targeted gene panel with a small set of genes and some non-aberrations, we could only match 18 to 38 or 39% of those patients. And those numbers, even so those genotype trials really show that we could boost the response rate, even make it like twice bigger, we still only can match a small subset of the population, which is very frustrating. We spend a lot of money to sequence, we have very high expectation, and still the majority of the patient will not benefit from those approaches. So there is definitely what we call a lack of biomarkers. We don't know enough about those molecular aberrations and how do they associate with drug response. And here, the resolution is too poor. This is not meant to be read. This is a paper trying to recapitulate the non-biomarkers. And the list is actually not that big. If you look at OncoKB, which was mentioned in the previous talk, we have clinically approved biomarker for around 40, 60 drugs. This is anti-cancer therapies. This is not much. You can imagine we have a portfolio of drugs that's much bigger than that. For the vast majority of the drugs, we just don't know what makes a tumor more sensitive to those drugs. So there is a need. Most biomarkers are univariate, like a single aberration. It could be a mutation, an amplification, or an overexpression, for instance. Very, very, very few biomarkers are multivariate, meaning that they are trying to combine information from multiple genomic regions or genes to come up with a good predictive association with drug response. This is an example in breast cancer. The Oncotype DX is a combination of 16 genes at the expression level. It's one of the few expression-based signatures in breast cancer. So the big issue now is, how do we expand our portfolio of biomarkers? How do we associate more drugs to molecular aberrations? How do we find more actionable aberrations in those patient tumors? It could be univariate or multivariate, but we need to expand our portfolio. So finding biomarkers is definitely very challenging, especially when it comes to drug response. And the reason is that you can use different kind of biological samples or materials to actually study and identify those biomarkers. So of course, you want to cure patients. So the place to start is probably to collect biopsies, and whether it's liquid biopsies or solid tumors, and try to provide them with multimodal omics technologies. You could look at mutation, copy number variation, methylation, expression, proteomics, name it, and try to look at an association between how the patient responded to the treatment versus the state of the tumor at diagnosis or after surgery. So the problem here is that most of the data extremely scarce. We don't have a lot of those clinical trials. They take a lot of money, a lot of time, a lot of resources to organize. There are many ethical issues. You can only study drugs that have been approved for clinical usage. So this is definitely the best data type to find biomarkers, but this is by far the smallest in terms of studies and numbers. You can only study a very small subset of drugs. A little bit below, you could, in terms of recapitulating the tumors, you could basically try to generate cancer models. And the beauty is that you kind of clone the tumors, either in animals or in dishes, and you can start screening those tumors with drugs. And note, this is really, really important because you can study the same patient tumor and screen them with many drugs and study the response to those different therapies. So this is really a paradigm shift compared to studying patients. Now you can really clone the tumor and study multiple things in parallel. So PDXs, patient-derived xenografts, are actually now model of choice to predict drug response empirically and find molecular biomarkers. The problem still is that it's a rather low throughput platform to study, to identify biomarkers. You can only test a few drugs on dozens of PDXs. It's actually very hard to do big screen on many drugs and many PDXs. It's still a very lengthy process. You still have ethical issues. You're dealing with animals. So it's definitely a very important and relevant cancer model to find biomarkers, but it's still quite limited. So on the other end of the spectrum, you have those immortalized cell lines or those in vitro and ex vivo models, short-term culture of tumor cells. You can really do high-throughput drug screening. We have robots to do that. They are easy to handle. It's relatively cheap to do so. You have almost no ethical issues. The big issue, so, is that they lack many features that are present in the patient tumors. They lack the microenvironment. There is no functioning immune system. They grow very fast. They tend to drift over time genetically. So there are a lot of drawbacks regarding those models. However, you get tons and tons of data. So I'll show you how you can use those data to find biomarkers. And then, once you find those biomarkers, the thing to do, and the usual pipeline, is you kind of organize preclinical trials in those in vivo models, and you finally test those biomarkers in the patient if they translate in vivo. So now you have an idea of the whole pipeline to find drug response biomarkers, starting from very simplistic model to more complicated preclinical models to finally try to apply and validate the clinical utility of that biomarker in the patient. So this is a field called pharmacogenomics, and that has many definitions. So here I mean in vitro drug screening with genomic profiling. So the way it works is that you have a large portfolio of cancer models, in that case immortalized cancer cell lines. Let's say you have 1,000 cancer cell lines. There are many more. You provide them with all the profiling, the genomics technologies you can imagine, DNA, copy number variation, methylation, expression, and so on and so forth, depending on your budget. Now you can really deeply characterize those cell lines at baseline before drug treatment. And then what you do is you treat those cell lines with drugs, and you try to quantify their sensitivity or their response to therapy. And as you can see here, the top right, this is what we call a drug dose response curve. So on the x-axis, you have different concentration of the compound, and on the y-axis, you have the viability of the cancer cells. So a drug that's particularly inefficient would just have an horizontal straight line at 100%, meaning that none of those cells actually die due to the treatment. A treatment that's much more efficient would have this kind of sigmoid curve where at some point, with a sufficiently high concentration, you start killing a lot of those cancer cells. And this inflection point is actually very important because it means that you can control the efficacy of the treatment by changing the concentration. If you get something really toxic that kills everything right away, this is probably not a good drug for the patient. So another goal is to really link those two kinds of data, genomics data and the drug sensitivity data. So one, I'm going to call them molecular genomic profiles, and the other one are pharmacological profiles. So you get your molecular profiles, could be a patient tumor set of mutations, for instance, or in that case, cell lines. You have your pharmacological profiles. You know how those cell lines respond to treatment. And what you want to find is this magical function f of x, which is basically the biomarker. You can predict what's going to happen to a cancer cell based on the molecular features of those cancer cells. We knew for a long time that genomics data are extremely noisy. So we talked a little bit about the quality of sequencing data, the DNA, which is probably the easiest thing to measure, and it's the most stable. RNA is actually more challenging. It's also time-dependent measurements of the state of the cell. If you do it at different temperature level, even in the lab, you may get a slightly different readout, so it's a little bit more sensitive. But here you can see that for gene expression, for instance, some genes are actually very, very robust. I took two studies here. I'm going to present those studies a bit later, but one from the Sanger and one from the Broad Institute. They profiled the same cell line with the same technology. Gene expression, microarray gene expression profiling. You can see that one gene nicely aligned with the diagonal, meaning that those two studies really agree very well on which cell line express which gene at which level. However, there are some genes that have a very nice spectrum of expression in one study and a very, very narrow expression space on the other study, and they are completely uncorrelated. So you have to be aware that sometimes the noise is so high that you cannot really trust the measurements themselves. So we knew that for genomics data for a long time. What we didn't know that much, at least on the bioinformatics machine learning side, even the clinical setting, the clinical side, is that the pharmacological data, actually the results of a very complex experimental protocol. And those pharmacological profiles could be extremely noisy as well. So here I give an example where you see those two curves that completely disagree between the two data sets. One data set said that this cell line is extremely resistant to the therapy. There is absolutely no sensitivity observed. And the other data set measured this very nice sensitivity. Even so, it was the same cell line with the same drug. So that's kind of annoying when you look at biomarkers. You know, you could build a very good biomarkers that works in one data set, but it will never work in the other data set just because the data disagree. It has nothing to do with the quality of the algorithm. This gold standard or truth is actually noisy on its own. So thankfully, we don't have one data set. If we had only one data set, we would be doomed because we would not really know what's robust to noise. But we have many of those data sets. So along the years, starting from the 90s with the NCI drug developmental program, they basically screen many drugs on many cell lines. And the beauty is that because they use immortalized cell lines, they kind of reuse the same cell lines again and again, the same way that they would investigate the same drugs again and again. So there is actually quite an interesting overlap across those studies that will allow us to do interesting things. However, when we have talked about curation during the first talk, and this is definitely a major issue here as well. After, I don't know, 80 years of research, nobody came up with a standard way to name those cell lines. Those drug compounds is really a mess. Everybody would use a different nomenclature, whether it's the Sanger or the Broad. And we are talking about excellent, really outstanding teams that are doing their best, and still they cannot figure out how to name those cell lines the same way. So we kind of had to take all those data sets and try to understand what are the synonyms of the cell lines that are being used across the data set. So we mapped it to Celosaurus, who's an interesting database with 10,000 cell lines or 20,000 cell lines now across all different organisms. So it's very, very comprehensive. And then we did the same thing for the drug compounds, and that's even worse. Sometimes you have the molecule name or the brand name or the approved name, so it's really a mess. And for a computer, when you compare AZD6244 and selumetinib, it doesn't look like it. So it's definitely a big issue, so we did as well. Now we can even, because we have the molecules, we can even do fuzzy matching, so we can identify molecules that are pretty close from a structural perspective. And because we didn't want to do it once, we didn't want to do it twice, sorry, so we did it once, we built a platform, and now we released it to the community. So everybody can basically download the created data set directly, which will save you probably 3 or 4 years of your life. So hopefully you don't have to suffer as much as we did. So in a nutshell, what it does is that we collected the genomics data, processed them with the basic the most established pipelines, and the problem is that when you have many of those pipelines, you know for sure that there is no consensus in the field yet, so it's not a solved problem. So here, we're trying to release data processed in many different pipelines, so you can always compare or test different processing pipelines. We did the same thing for the cell viability data, trying to define all those dose response curves is actually pretty challenging. Excel is definitely not a good tool to do this. We had to write a bunch of script to make sure it's fully reproducible. And then we use our semi-automated creation pipeline to actually rename the cell lines of the drugs and even the molecular features. So we have a better overlap across the data set. And it's getting easier over time because human tends to do the same typos in the cell line name. So it's actually pretty convenient. And the beauty now is that you don't have to do that anymore. You can download what we call a pharmacoset. It's a special type of object that contains everything, including all the curation. So it's very convenient. You can download that data set and we have a series of analytical functions that does very basic stuff like fitting the drug dose response curves, summarizing the sensitivity to one number. So like pharmacologists like to use the IC50, for instance, the concentration you need to kill 50% of the cells. We have the same thing to merge replicates among omics data. And so now you can really extract the information you need to do biomarker discovery quite easily. But this is for people with a set of computational skills, which is not always the case. Or even myself, I don't always want to open a terminal and start typing code. So we created what we call PharmacoDB. It's a web application that you can use to actually mine all those data sets very easily. So let's say if you type a cell line name, what you get is all the metadata we could collect about that cell line, as well as a link to the external databases describing that cell line. So this is an example of a lung cancer cell lines, and you can see on the right all the data set where that cell line has been used. So it's now very easy if you have a cell line of interest to actually extract all the pharmacogenomics data that you need. You can do the same thing with a drug, like AZD6244 or selumetinib, and you can, again, look at all those data sets that have actually profiled or screened that drugs, again, a bunch of cell lines. More interestingly, if you type a cell line name and a drug name, what you obtain is all the drug dose response curves that we have. And this is very typical of the kind of data you get at the end of the day, where there is a huge spectrum. So if you look at the different data sets, you would see that GDC 1000 seems to have this curve more reflecting a very high sensitivity, while the other data sets tends to say that this cell line is only moderately sensitive to the drug. And you have a whole spectrum. But even within the same study, you can look at the two orange lines. Those are two biological replicates done at different sites, one in Boston, one in UK, and you can see that they vastly disagree. So this is something really common in those data sets, and that's where having multiple data really helps you to see whether there is a consensus in the field about the sensitivity of the cell line versus against a drug. And those curves are kind of complex in a way, so many times you just want to summarize them into one number. So here, we computed an array of statistics that you could use, whether it's the area above the curve, higher means more sensitivity, or the IC50 already described, or a bunch of other statistics that you can use. That has been published last year in Nucleic Acid Research if you're interested. So the beauty now is that after all this work of curation, now we can look at overlaps across data set. And this is an upset plot. I don't know if you ever saw a Venn diagram with more than 7 circles. It's actually quite hard to interpret, so they came up with that new way to look at overlap across data set. And here you can see, for instance, that there are like 300 cell lines that have been screened in 4 studies. This kind of overlap rarely happens any times in biomedical research. But here, because they screen the same cell lines or the same drug all the time, you see you have a lot of replicates of the experiment, which is very interesting. And if you look at the drugs, the overlap is much smaller, but you can find 7 drugs, for instance, that have been screened in 5 data sets. So you still have quite a lot of data to work with. So here, we published a quite infamous paper in Nature in 2013 where we basically compare 2 famous data sets and say that they were not consistent. And we got a lot of heat for that, so that made us thinking about, how do we really need to look at those data? And there have been a comment about looking at the data, and I think this is quite important. And we designed a way to not only look at the concurrence across data sets, but also find new biomarkers in a way that takes into account the noise in the pharmacological assay. So what any pharmacologist would tell you is that if you have an IC50 of, let's say, 1 micromolar or 10 micromolar, it may not matter because this is kind of the variation you see when you repeat an experiment. So why would you try to compare cell lines that are like 1 micromolar and 2 micromolar? You're just in the noise region of the assay, so there is no point comparing those cell lines. So this is what we've done here. We kind of modified a statistic called the concurrence index, where you compare every pair of cell lines and you see how many times you rank them the same way. So a perfect classifier would always tell you that the right order of the cell lines, that this one is more sensitive than this other one. And here we basically discarded all those pairs of cell lines that are very, very close by sensitivity data. And you can see that usually in the field, concurrence index of 0.8 is very good. You can see that many of the drugs now have a concurrence index of 0.8 when you start to discard those pairs that are irrelevant due to the noise in the assay. So now we have this analytical way to basically integrate those data sets in a better way. And when you do that, we looked at all the clinically approved biomarkers for those drugs. So in an ideal world, you would find all those biomarkers to be significantly predictive of drug response in vitro. However, you see that there is a bunch of no there. There is a bunch of data of biomarkers that could only be found in one data set but not the other, or in none of the data set. And that's due to many factors. It could be that the drug mechanism of action kind of interfere with the way you measure cell viability, whether you measure the amount of DNA or the ATP generated by the cells. So that matters. It could be that the set of cell lines is different. So you may look at an amplification that is truly associated with drug response, but you were unlocking your selection of the cell lines, and only 2 cell lines out of the 50 in the other data set got that amplification, so you don't have the statistical power. So there are many reasons why you don't find all those biomarkers, but there are still many biomarkers that you can find. So those preclinical data actually could find clinically relevant biomarkers, but not all the time. And because we have all those data sets now, you can easily go online, go on PharmacoDB, download the data, and do something like this. This is called a forest plot. It's a meta-analysis where you basically look at the association between here ERBB2 expression with a drug called lapatinib, which is an ER2 inhibitor. So these are non-biomarkers. This is why I show it. It works. You can see that in every data set, you get this MCI that's much bigger than 0.5, 0.5 being the rundown case. And that just means that if that gene is highly expressed, the cell lines will be highly sensitive to that drug. And interestingly, you can also see that 2 data sets kind of disagree. One seems to say that the effect size, the quality of your biomarker is very, very low. And that's CTRPV2, and that's because they screen a very small number of breast cancer cell lines. That's where ERBB2 got expressed most of the time. The other data set, gray, had this beautiful effect size. Very, very high MCI, modified concurrence index. And that's due to the fact that this is the largest set of breast cancer cell lines that have been ever screened today. So you kind of understand what makes data set different by just looking at the results and going back to the metadata and try to understand how those studies has been designed. we also wanted to look at different classes of biomarkers. So as I showed before, most biomarkers are either protein-based or DNA-based or expression-based. But when they look at expression, they usually look at the overall gene expression. They kind of ignore this information about all the different versions of the mRNA that could be expressed. And here we decided to take RNA-seq data the same way that Adam did. Instead of looking at gene fusion, we actually look at different isoform expression, which is actually a quite difficult problem. But we found that those isoforms are quite interesting. And how we did it, because it's definitely a challenging issue, we kind of leverage all the data we had to find robust biomarkers. So first, we did what we call a meta-analysis. So we fused two data sets using different pharmacological assays. And the idea behind was to find biomarkers that would work whatever the assay you use. So it would be kind of robust to that experimental factor. And then what we did was a pan-cancer validation using a completely independent data set released by Genentech years later. So now we kind of have a set of biomarkers that seems to work in multiple data sets on a pan-cancer basis. And we also had two breast cancer specific data sets that we leverage, including a new one generated especially for that paper, where we have several rounds of validation to make sure that our biomarkers are very robust. And in a nutshell, what we found is that the isoform biomarkers in red, so the gray bar shows the total number of potential candidate biomarkers we found in the training set, those two data sets we use in the meta-analysis. And I'm not sure if you know the concept of overfitting, but the point is that when you have a lot of different things to test and you have a limited amount of data, it's actually very easy to find biomarkers that seem to do extremely well in your training set. If you're not careful, this is just an artifact in the dataset. It will never reproduce in other dataset. It's just due to the fact that you tested so many things randomly, you're going to find a bunch of stuff that work very well. So here, that's why we really needed so many independent datasets. So here in gray, you have all those candidate biomarkers. We know that many of them are wrong, and the only way to figure out is to test it in another dataset. But you can see that the isoforms have a much larger proportion of validation than overall gene expression, which is kind of interesting. So those isoform expression not only seems to be more predictive of drug response, but it also seems to generalize better. So I will pass that slide. I will just say that somehow you want to build multivariate biomarkers. For many drugs, you could not find a single molecular aberration that's predictive of drug response. And that's due to the fact that cells are basically governed by networks of pathways that interact with each other. So it's actually not so frequent to have one molecular aberration that seems to be predictive. Sometimes you have to go multivariate and consider the whole context of that cells. Is it like there are other mutations that you have to take into account, methylation patterns or expression programs? But those things could go quite complex, and finding multivariate biomarkers is actually a very difficult task. We actually look at EMT genes, for instance, and the reason we looked at EMT genes is because at the expression level, they're beautifully simple. You have this bimodal expression, so you have two biological states that you can easily discriminate. And when you do that, you get these binary features, whether the cell line is in state 1 or state 2. And once you got this, then you can build very easy formulas such as that one, where all those genes are EMT-related genes, and you basically say whether VIM is expressed, or if CDH1 is not expressed, or this, or this, or this, then you get a better response to statin. And this is a very overly simplistic multivariate biomarker, but this is the kind of biomarkers we would like to bring to the clinicians, because they can look at them, they can try to understand them, and see whether it's related to any of the mechanisms of action known for the therapies. As I said, we would like to bring those biomarkers to the clinicians, and that's really where there is a big gap. People do awesome preclinical research, people do awesome clinical research, but don't ask me why. It looks like the communication is broken. The clinicians will not trust what the preclinical models tell them. On the other hand, the basic researchers cannot use millions of different clinical samples to study any kind of drugs. So we wanted to basically close that gap by trying to bring those preclinical biomarkers that are probably wrong because they're based on preclinical models, but they're still a very valuable source of information very close to the clinicians. And the way we did it, it has been cited several times, we want to use cBioPortal, which is a great web application where you can investigate the molecular aberrations of a single patient or population of patients. And because it's now being used more and more by clinical researchers all over the world, we thought that that would be a very good interface to show our preclinical biomarkers. So the way it's going to work is that usually you get this page where you see all the molecular aberrations of the patients. And here you see amplifications in many genes. You have this OncoKB icon that clinicians like to look at because those are clinically approved biomarkers. So if you have an ERBB2 amplification, OncoKB will tell you, well, you should probably prescribe lapatinib for that patient because here it's a male breast cancer patient. You get the ERBB2 amplification. Lapatinib has been approved for breast cancer, so go ahead and use it. But for many patients, as I said, more than 50%, you don't have any of those actionable mutations. So for those patients, you can have this little pill that will be basically reporting the associations that we can find in immortalized cancer cell lines with all the drawbacks that it entails. So in that case, of course, you're going to find lapatinib. That's a non-ERBB2 inhibitor, but you could also find other compounds like the CP72 and so on and so forth, where actually it has been already tested in clinical trials, so the clinicians could feel comfortable to actually order the drug and prescribe. But at least it provides the clinicians with a new data stream to a new set of evidence to basically potentially influence the treatment decision process. So I won't go into the details, but as I said, once you have a preclinical biomarkers in vitro, you would like to validate it in vivo. However, if you thought it was hard in vitro to find biomarkers, it's even harder in vivo. Those experiments are extremely hard to do. Those curves are extremely noisy. You need a lot of replicates to really understand what's going on. So you have to expand your population of mice. You have to treat them with 2 arms, et cetera, et cetera. So it's a very, very similar concept, but the data are actually even harder to gather. And we are working on a platform similar to what I've shown before for in vitro, but for in vivo drug screening. So the problem as well is that you have very different designs for in vivo drug screening. I won't go into the details. But to conclude, I hope I convinced you that not only we have tons of data on the preclinical side to study this association between molecular aberrations and drugs. And those data have been generated to understand what the drugs are doing, but also to understand what's the subset of population for which we should use those drugs. So we designed PharmacoDB, and we try to integrate it with cBioPortal so everybody can really use them, use those resources, not only the bioinformaticians, but also the clinicians and the biologists. We try to close the gap between not only the preclinical side and clinical side, but also ideally we would like to combine in vivo data and in vitro data to find better biomarkers and really to try to increase the translational potential of those biomarkers by taking into account all the kind of data we could gather for those drugs and patients. And as I said, we really make a point at making all those resources not only open source, publicly available, easily to access, well documented. And the reason is that I'm sure there are tons of errors in what we do. Every day we receive feedback from users saying, you know what, you claim that you have curated that cell line, but I found that typo in that cell line, and I'm sure that that means that. And the problem is that we have to deal with over 1 million drug dose response curves with many, many data. Of course, we're going to miss a lot of things. And the beauty is that if you share and if you put into place some kind of feedback mechanism, we can actually learn from the users as well. You're not supposed to be passively using those tools. You're supposed to report what worked and what didn't work. So here we try to put those mechanisms into place. For instance, we try to engage the clinicians to tell us what are the in vitro biomarkers that seems to work or not work in clinical setting. Once we know that, we could probably change our pipeline or update our pipeline to actually get better biomarkers in the future iterations. So we're really committed to this. So as I said, we share the data, the metadata, the code. We share the packages with documentation. And something that we don't really consider that much nowadays is that it's actually hard to use a tool. It sounds simple, but most tools are actually complicated to install and to use. And the reason is that sometimes you run an analysis on, let's say, a Windows machine with a given version of software XYZ. And that gives you a set of results. And you give exactly the same data to somebody else, and they're going to use maybe a Linux machine with slightly different software libraries. And that may actually impact the results. Hopefully not in a meaningful way or in an impactful way, but sometimes it could really go wrong. So what we try to do is to share what we call a virtual machines where everything is self-contained and installed, so you can download that virtual machine, run the same code or the same data exactly, and get exactly the same kind of results. And these things is very, very time-consuming. From a pure academic career perspective, this is suicide. Nobody would ever value this kind of stuff, except if you think in terms of science and impact. Because now people can actually take your resources and adapt it to do something else. Or you can take it and make it better. So this is not about you anymore. It's about the whole scientific community. But it has some good consequences. So when you have a new member coming to your lab, it's actually way easier for that new member to learn and to reproduce analysis. When you have a paper revision 6 months after you submit it, but a postdoc left, usually that's really painful. You cannot even address all the reviewer comments. But with that kind of approach, it's definitely easier for anybody to take, reuse, adapt, reproduce. So with that, I would like to thank you, a lot of my colleagues and the funding agency. Thank you.
Well, that was really, I like your attitude.
That put me in trouble sometimes.
Any questions from the floor? I have lots of questions, but this is really interesting stuff. I think this is amazing work, all three speakers, what they told us today. I mean, we're very lucky to have such quality people here. The idea of farming pharmacological data from cell lines, to be honest with you, Benjamin, I would, if you had said that you were going to do this, I'd say you're out of your mind. Mm. Because, as a wet lab scientist, you can have a student do an IC50 on a particular set of cell lines with this drug in January and do it again in August, and the data is not going to be the same. Because the passage number of the cells might be different, the age, how old the drug is sitting in your freezer may be different, and so on. So it's a monumental task. I think what you've done is really amazing. And I'm sorry to say, I wasn't aware of pharmaco-CB, and that is bad.
It's not a secret resource.
No, but that is bad. So
but
I'm going to change that.
But the funny thing is that a lot of people have this same. So when you generate the data, you know exactly the limitation of the data. You know what kind of protocol you use. You know what kind of noise you expect. But then you have another part of users who's just going to take it for granted that those are good data.
Yeah.
Right? And so people keep reanalyzing the same data set without really knowing the limitations of those. And honestly, when we published our paper comparing the different data sets, we were basically the most naive pharmacogenomicists on Earth. We thought that those were like good data, and we were shocked to see that some cell lines could change their sensitivity status from one data set to the other. So half of the world were shocked by the results. The other half say, of course.
Yeah.
Like,
I would have said,
I know those data are very noisy, but guess what? You never wrote this in a Nature paper, did you? Right?
But.
So that's a problem of communication and expectation.
One of the things I'm a little bit concerned about what you said, okay, there's lots of noise. You said, okay, 1 micromolar versus 10 micromolar for an IC50. For me, on the lab bench, that could be significant. So how did you determine what is noise? What are the parameters of noise? So that you can filter down and get real data.
Yeah, I didn't show that slide, but when you have so for some data set, you can do a lot of replicates, and you have to be the Broad or the Sanger Institute to do that. You need a lot of money. Yeah. You need a very large screen. When you do enough replicates, you start to understand what kind of differences between two drug dose response curve you can trust. And if you look at different data sets, you're going to have very different noise distribution. Mm-hmm. But if you can model that, then suddenly you know what are the changes that are truly biologically relevant versus changes that could be completely artifactual, just due to technical noise or biological noise. Once you understand that, and once you have access to many data sets, you can look at two things. You can first develop method that better takes into account the noise in the data. Second, you acknowledge that there is not a single protocol to generate the ultimate high-quality data. There are many of those, but your biomarker, if you want it to work in the patient, if it doesn't work between two in vitro data sets, you're kind of doomed to fail.
Yeah.
So you basically integrate all those data sets and find biomarkers that are very robust to all those sources of variation.
So it's all about stringency, basically.
It's
the amount of data and being really stringent so that you get great confidence on a particular
and also the experimental design. You have to spend some of your budget to do replicates to make sure that you understand.
So for the wet lab scientists, you know, what I'm hearing from you is that there should be an SOP for everyone doing IC50s for any particular drug. Everybody should do it the same way, the same cell lines, the same media, the same temperature, the same time period, the same source of drug.
But then you're going to end up with exactly the same problems every time.
Exactly.
But how? So what you're saying?
The more variation in experimental protocols that you've got, but that give you the same answer,
So
then you know it's not the protocol.
Okay.
It's
Yes.
the drug and the
Very
drug.
good point, Greg. Very good point. Thank you. I feel vindicated to
Well, it's
Actually-
the beauty
What
of
we do is
You have two. You have two levels. One, you need to understand the noise in the assay, and you have to minimize it.
Yeah.
And that's the boring part, where you do the same thing again and again. But that's still going to be biased. So that's where Greg's point is very important. You don't need one assay. You need multiple assays. And you need to see
different types of assays or the same assay done in different places at different times.
Phase one is what you just said. Understand the noise of the assay is not just technical replicates, biological replicates, different sources.
Okay.
So if you look at the MAQC, this group, that's what they do. They basically take a technology, they spread it around different labs all over the world, they try to have different SOPs, different ways to do it, and they compare intra-lab, inter-lab reproducibility.
Variation, yeah.
Phase two is really, it's all about the biology. For any given drug, any given cell line, one assay could be more biologically relevant than the other assay. But that's very, very hard to know.
Yeah, ahead of time, there's no way to know.
Exactly. And nobody has the budget to run do it 10 times. So
So
you really need, that's where and that resonates with one of the comments, one of the other speakers saying, look, we have a massive amount of data available. This redundancy in science is actually very important. You should share so people can mine. And once they start mining sufficiently large number of data, then suddenly you can see some patterns appearing that would be impossible to identify with a single data set.
So
It's-
that's reassuring.
It's interesting because this is a great discussion because qualitative science, I mean, it used to be said that you can't measure personality, that there's too much variation and too much noise. But I mean, the psychologists and the things that have been established to depict those differences are real. It's not necessarily as accurate. And we like to think of science as being really accurate, but we see that depending on the assay and the variability. But if we develop these bioinformatic approaches that can sort out the noise and find the commonality, then we can find meaningful differences. Is I think what in part you're saying, if I interpret that correctly.
Yeah, pharmacogenomicists would claim they're more quantitative, but after all the data I've seen, I think psychology is pretty good too. Yeah.
Well, that's
Adam,
great. I think, is there another question?
No, I wanted to ask Adam, so Benjamin is talking about using thousands of cell lines and all this, these preclinical type studies. You exclusively, in the Cell paper, used 80,000 tumor samples, a small number of pediatric and more adults. Why didn't you look at cell lines too in your study, or was that too many, or you?
We should collaborate.
We will look together. The cell lines didn't exist in those numbers at that point in time.
So they didn't really add anything.
No, and there's just such interesting variability in the human tumors that we, at the time, to be honest, didn't feel the necessity to look at the genetics of cell lines.
Okay.
But it's a little bit of a different experiment than what what's been described here. In terms of looking at the genomic landscapes, without perturbing them, I find that human samples are probably the most important.
Sure.
Experiments in nature, and they tell you what's important in that particular context. But they're always an N of one, you never replace.
Right, I mean, there's always this pull-push thing in between these preclinical models not ever being sufficient. But then you look at human tumors, that's a snapshot in time.
Yeah, and we can, and we have it after multiple rounds of therapy.
So it's the best we can do. And I think the key, if I've learned anything this afternoon, is that be friends with bioinformaticians. They are extremely important scientists. And going forward in medicine today, you will not be able to do anything without good bioinformatics.
And if I can make a little bit more enemies today, there have been a series of editorial in the New England Journal of Medicine highlighting a very interesting fact. So clinicians are very close to the patient. They put a lot of time, energy, and brain to collect the clinical samples, organize clinical trials, so they feel, they have this feeling of ownership regarding the data. The bioinformaticians are the opposite. They don't generate the data. They always complain about the quality of the data. And they always think it's very easy to generate data, okay? So we have to meet in the middle. Clinicians don't own the data. If any, it would be the patient, right? But we have to acknowledge that there is an insane amount of work to collect high-quality data. And that comes from identifying the patients, collecting the samples, but also finding the money.
Forget about that, just getting the research ethic.
Yeah, and the REB and everything. It's very, very complex. On the other hand, if you don't share, you don't push the science forward enough. But the bioinformaticians can do great things when they have data and they understand the data. So we basically need to find some kind of common ground where we both acknowledge the contribution of everyone and give credits to everyone. But for the moment, there is a bit of this dichotomy that doesn't really exist in the preclinical setting, but that really exists in the clinical setting. And Princess Margaret is pretty good at this. I mean, we have clinicians really sharing a lot of data. But if we build that bridge between more basic research and clinical research, I think we're going to understand each other a little bit better and share more. And that's going to be all beneficial for science and the patients.
Well, I think that's a perfect way to end this session. Thank you very much, Ben.